All right, so this lesson is all on growth of plants and the hormones that you're going to see within the plants. So for this lesson, these are the expectations that you need to know. So let's take a look. So one, undifferentiated cells in the meristems of plants allow interdeterminate growth. Mitosis and cell division in the shoot apex provide cells needed for extension of the stem and development of the leaves. Plant hormones control growth in the shoot apex. Plant shoots respond to the environment by tropisms. Auxin is the only named hormone that is expected that you need to know how to talk about. Auxin efflux pumps can set up concentration gradients of auxin in plant tissues. Auxin influences cell growth rates by changing the pattern of gene expression. Micropropagation of plants using tissue from the shoot apex, nutrient agar gels, and growth hormones. And lastly, use of micropropagation for rapid bulking up of new varieties, production of virus-free strains of existing varieties, and propagation of orchids and other rare species. All right, so you might have already taken a look at meristematic tissue. So plants will grow at the meristem. So meristem is an area within the plant where you have undifferentiated cells. So they're essentially like the stem cells. And these cells are going to divide through mitosis and allow the plant to grow. So they can be found an apical, which is the root or stem tip, or lateral, which causes thickening. So growth of a multicellular organism, such as a plant, is produced by either one or of the combination of cell enlargement or increase in the number of cells through division. So let's look at the apical meristem. So the apical meristem, once again, can be found in two locations. We have the shoot apical meristem, which is found at the tip of the plant. So tip right here. And we can also find it in the root tip of the plant. So you can imagine that the apical meristem and the shoot is going to cause it to grow upwards, whereas the root is going to grow downwards. So this vertical growth we call primary growth. And primary growth occurs in herbaceous, non-woody stems and roots. So herbaceous would be plants that don't really have bark on the outside. And then eventually, as these cells divide, they will become differentiated and actually become specific types of cells. So in the stem, growth occurs in sections called nodes. So we can see that we do see branches, and where these branches forms, there are nodes where you do have these undifferentiated meristematic tissue. And eventually they will grow to become branches. So let's take a look at the different hormones. So we know that hormones are chemicals that can be transported to another location and cause physiological activity, such as growth and metabolism. So the main hormone that you definitely need to know is auxins. So auxins is the main hormone that's going to cause growth in a plant. But there's tons of other functions, including root and shoot growth, flowering, fruit development, leaf development, and wound response. Cytokinins are a hormone produced in the root which promote auxiliary bud growth. And as you can imagine, cytokinins are going to promote cytokinesis and cell division. There's gibberellins that are hormones that contribute to cell elongation, and we're also going to learn about this when we talk about germination. And lastly, we have this IAA, also known as acetic acid, and it's going to play a role in cell elongation, but also in the opening and closing of stomata. All right, so going back to primary growth, we have this idea of apical dominance. So essentially what this means is the plant wants to grow as high as possible. That way it can get closer to the sun and therefore be able to perform more photosynthesis. And so what happens is auxin in the apical meristem is going to cause this growth. But something that's interesting is that auxin actually inhibits the growth of branches. So as the shoot starts to grow upwards, the apical meristem starts to get further from these nodes. And once the apical meristem is far enough away, which is where the majority of the auxin is going to be, once it's far enough away from these nodes, that's when they have the ability to branch out. So this is called apical dominance because it's focusing on growing upwards before it starts growing branches. So once again, remember we have these nodes that can eventually form branches, and when auxin is nearby, it actually inhibits the growth of these branches. But as the plant grows upwards, the apical meristem is far enough away from these nodes that they are no longer inhibited, and that's when we can start to see branches form. Now, if you were to cut the apical meristem of a plant, then you no longer have that auxin that's going to inhibit the nodes, and therefore you're going to see a lot of branching, and it's not going to be able to grow upwards past that point. When we have lateral meristem, there's kind of two what we call areas of growth. We have our vascular cambium and our cork cambium. And once again, the lateral meristem is going to cause what we call secondary growth, which essentially causes the thickening of the plant. Because as the plant grows upwards, you're going to want it to get thicker so that it doesn't flop over and it has the strength to maintain itself. So the vascular cambium lies between the xylem and the phloem, and this is going to, these differentiated cells are going to eventually become new vascular tissue. So as it starts to grow in thickness, you want to make sure that there is vascular tissue to be able to transport water and nutrients. So the vascular cambium is going to allow it to create more xylem and phloem so that it can transport these nutrients. The cork cambium is more on the outside, and this is going to create more cork cells, and it essentially creates the outer bark. So this would be for woody plants. You do not need to know this, but this is just showing that kind of the level of differentiation of these tissues. So cells in the meristem are continuously producing new cells by mitosis and cytokinesis. The root apical meristem is responsible for growth of the root, whereas the shoot apical meristem is responsible for the growth of stems, leaves, and flowers. So during each cell division, one cell remains in the meristem while the other increases in size and differentiates as it pushes away from the meristem area. The apical meristem can give rise to additional meristems, the protoderm, procambium, and the ground meristem. The protoderm develops into the epidermis, the procambium eventually becomes vascular tissue, and the ground meristem will give rise to the pith. Once again, you do not need to know this. And it's just showing once again how the different layers form, and you could see that this is what actually causes the growth rings in trees. So here we are, growth rings. So the rate of secondary growth will change depending on the season, and if you can recall from grade 9, the grade 10, this was one of the tools that we can use to look at what the weather patterns were like in the past. So the thicker the ring meant that the tree grew more that year, and that probably means that the conditions were more favorable. So this whole idea of looking at these rings is called dendrochronology, and the rings represent a year of growth. So you can count how many rings there are, and that's going to tell you how old the tree is. Okay, so tropisms. So tropism is just a plant response to a directional stimuli. So there's lots of different tropisms. The main one that you need to know is phototropism and gravitropism. And we can have positive and negative tropism. So a positive tropism would be growth towards a stimulus, whereas negative would be growth away from a stimulus. So this is a very popular paper 2 question explaining how phototropism works. So phototropism is when you notice that if you've seen any time-lapse videos, the plants tend to follow the sun. They tend to grow towards the sun. And you need to, so that is a directional growth response. It's growing towards the light. So you need to make sure that you can explain how this works. So first off, you need to know that auxins promote the lengthening of cells, not division. They cause cells to grow in length. And they can be found in this thing called the coleoptile, and it's a protective sheath around the emerging root or shoot. And once again, this auxin is mainly found in the apical meristem. So under normal conditions, auxin is distributed evenly around the shoot, causing it to grow upwards. However, there are photoreceptors in this coleoptile, and it detects light, a light stimulus from one direction. So if the sun was over here, the light would be hitting the plant in this direction. What ends up happening is auxin it will be transported to the opposite side, or the shaded side of the plant. Now we have an uneven distribution of auxin. So obviously the side that has auxin you're going to see more cell elongation, whereas the side that does not have auxin, you're not going to see that. So as a result, we see that the auxin, it elongates the one side and the other one stays. So this causes the stem to actually bend towards the light. Okay. And we'll talk about gravitropism, aka geotropism, and how it works. But let me just play these videos for you so that you can kind of have a sense what I'm talking about.
If a plant has enough water, minerals, and energy, it will grow, right? Well, sort of. But there is more to it, like why do plants bend towards the light, and not just grow straight? And why is it that the stem grows up, but the roots grow down? It isn't as if the plant has eyes to tell it where the sun is. You should already know about tropisms, so phototropism and geotropism. But if you have forgotten, watch our video called Plant Hormones, Tropisms, first. But how does it actually do this? Having been made in the tips of the stems and roots, auxin is moved in solution by diffusion to older parts of the plant. In the stem, the auxin causes the cells to change in elasticity. More elastic cells absorb more water and can grow longer. Strangely though, stems and roots respond differently to high concentrations of auxin. Whilst the stem cells grow more, the root cells actually grow less. So auxins make plants grow. But why do they bend towards the light? How do they know to do this when they don't have eyes? The bending happens because the light hits on one side more and breaks down the auxins on that side of the stem. So then growth slows down on the light side. The faster growth on the dark side causes the shoots and leaves to turn towards the light, which is ideal for the plant for photosynthesis. Auxin is produced in the tips of growing shoots. If the tips are cut off, then no auxin can be produced and so no plant growth. If the tips are covered, whilst auxin is still produced, light cannot break it down, and so phototropism cannot occur. The plant just grows straight up and does not bend towards the light. Pretty straightforward so far. Now let's switch it up a bit. Auxins have the opposite effect on root cells. In roots, auxins cause less growth. The shaded side of roots contain more auxin, and so they grow less. This enables the light side of the roots to grow more and bend away from the light. And if that wasn't weird enough, we have the opposite happening with auxins to gravity too. In a horizontal root, the bottom side contains more auxin and grows less. So the root bends downwards in the direction of gravity. So positive geotropism. But of course, the stem responds differently. In a horizontal stem, again, the bottom side contains more auxins because it is less directly hit by sunlight. But because auxins cause growth in stems, the bottom side grows more, causing the stem to bend upwards, against the direction of gravity, so negative geotropism. But I'm giving auxins too much credit. They don't work alone. They have a partner in crime. Cytokinins. You don't need to know anything about these hormones, other than the fact that they work alongside auxins. There is another plant hormone, however, that you do need to be aware of. Gibberellins. Once a seed germinates, the roots and shoots start to grow. But for this, the seed needs energy. Luckily, the seed releases a hormone called gibberellin, which causes the starch in the seed to turn into sugars and provides the seed with energy to grow. As well as causing shoot growth, gibberellins can also stimulate flowering and the production of fruit in some plants, and they also work with auxins to cause stem elongation. So now we know all about how plant hormones work. Have a think about how they could be used. We will discover some uses in our video on how we use plant hormones.
All right, so it kind of talked about the gravitropism and gave you a little intro to gibberellins and how it causes seed germination. Here's just a quick little video just to show you a time lapse throughout the day and how you can see the plants actually move in response to light. You can see as the sun moves, you see the plants start to grow towards the sun. Okay, so how does auxin actually affect gene expression? So proteins called phototropins absorb light of certain wavelength and change the shape so they can bind to receptors which control transcription of specific genes. So the genes involved seem to code for a group of glycoproteins that transport auxin from cell to cell. These proteins are called PIN3 proteins. So these PIN3 proteins can vary in position and type and therefore control the transport of auxins to where it is needed. So cells contain an auxin receptor. When auxin binds to the receptor, transcription of specific genes is promoted, and the expression of these genes causes secretion of hydrogen ions into the cell walls. The hydrogen ions will loosen the connections between the cellulose fibers, allowing cell expansion. So I haven't really seen this whole PIN3 proteins ever in a question, but you do need to know when you are describing phototropism, like when you talk about what auxin does. So I mentioned before that it causes elongation of the cell. Well, it does this by, once it binds to a receptor, it's going to cause transcription of genes. And these proteins that are produced are going to cause the secretion of hydrogen ions into the cell wall, which essentially kind of makes it more loose and flexible, and this is what's going to allow the cells to elongate. So gravitropism, as you heard, is kind of the opposite. So this is what causes roots to grow downwards in the direction of gravity. So in the video, it mentioned that in the roots, auxin actually has the opposite effect. It prevents cell growth. And so what happens is, and you saw if there was a horizontal root, you notice that the auxins will tend to be more concentrated at the bottom. And once again, this is going to inhibit growth at the bottom of the root, causing elongation at the top. And as a result, you will notice that it will bend downwards. So the concentration gradients of auxin are necessary to control the direction of plant growth. And once again, this whole idea that auxin needs to be unevenly transported amongst plant tissues in order for it to grow towards the light. So how does auxin enter the cell? By diffusion in these influx transporter proteins. I've never seen questions on this, but you never know. So it says different factors can affect transporter proteins and hence the direction in which auxin can move. So the location of the transporter proteins can be changed as the plasma membrane is fluid. So efflux transporters can congregate at the top of a cell in the roots to move auxin upwards. Transporter proteins can be activated or inhibited by stimuli such as light. So once again, I've never really seen any questions on this, but you can see that it is actually an expectation that you do need to know. All right, getting to the end. One way that we can make lots of plants is through a process called micropropagation. And essentially all that it really does is it takes undifferentiated mast cells in this area called the callus. So kind of like part of the meristematic tissue. And you will put it into these cells into like an agar plate, and then you're going to include hormones such as auxins and cytokinins. And this is going to promote growth of the plant. You don't need to know any of these like ratios, but this ratio of auxin to cytokinin is typically around 10 to 1, and roots begin to develop. Shoot growth media ratio of auxin to cytokinin is less than 10 to 1, and shoots begin to develop. So once again, we're taking these undifferentiated cells, putting them into an agar plate that will have nutrients. And we're also going to include hormones in there, such as auxin and cytokinins, to allow these to grow, and eventually they will become new cells. And then once they start to grow, we can eventually then transport them and put them into soil. So what is the benefit of doing this? So we can do this for rapid bulking up. This is just one way we can make lots of different plants. And another great thing about doing this method is that you know there aren't going to be any viruses in the plants, because typically viruses are transported in the vascular tissue via the plasmodesmata. The meristematic tissue does not have vascular tissue, so therefore if we take these cells, we know that viruses will not be in them, and so when we create these new plants, they will be virus-free. So a very common plant that we use micropropagation is orchids. They're actually quite difficult to fertilize and pollinate. So it's very difficult to get these orchids to breed sexually and maintain desired traits. So as a result, a lot of orchids are created through this micropropagation. And then what you can also do is when we have these meristematic tissue, we can do something called cryopreservation, which is essentially just like a seed bank. We can freeze these undifferentiated cells using liquid nitrogen, and then we can always go and unfreeze them and create new plants if we need. So obviously plant interactions are between different plant hormones can be quite complex, and our understanding of this area of science is steadily growing due to our ability to detect trace in small amounts of molecules. So here is a summary of all the different plants hormones. So I do want to talk more about the abscisic acid, because I've seen this as a paper 1 question. But let's just review. So first off, auxin. Auxin is the main hormone you definitely need to know, and it promotes primary growth, lengthening by promoting cell elongation and increasing the rate of cell division. And remember, it does this by pumping hydrogen ions into the cell walls, which kind of loosens up the cellulose and allows the membranes to be more flexible, and allows elongation. It promotes apical dominance, where the apex or the tip of a plant grows while the lateral buds remain undeveloped, because once again, the auxin will actually inhibit growth of these nodes, and it's not until it's far enough away from these nodes that the branches can eventually start to grow. Auxin concentrations may change in response to directional stimuli, so we know this through phototropism. Gibberellins triggered germination in dormant seeds, which we will look at in the next lesson. Gibberellins also causes stem elongation and promoting cell elongation and cell division as well. So if you've ever seen lettuce, sometimes what you can see with lettuce is it can bolt, where it just shoots upwards, and this will be caused by this hormone called gibberellins. Cytokinins promote cell division through cytokinesis and ensure roots and shoots grow at equal rates. It promotes secondary growth and the thickening of a plant and control the rate of branching by a plant. Cytokinins are also involved in stimulating the growth of fruit. Okay, abscisic acid, or ABA, is kind of a stress hormone. It functions to inhibit plant growth and development. It promotes the death of leaves, this is called abscission, and is responsible for seed dormancy. So in the wintertime or in the fall, when it starts to get colder, this is where you're going to see a lot of abscisic acid. It's kind of the stress response and causes the death of leaves. So it generally initiates stress response in plants, like winter dormancy and deciduous plants. It controls the closing of stomata and hence regulates water loss in plants. So if it closes the stomata, you definitely need to know that abscisic acid closes the stomata. And the way that it's going to do this is cause water to flow out of the stomata. When water flows out of the stomata or the guard cells, they become less turgid, and then the stomata can close. And if the stomata are closed, then there's no more gases being able to move in and out, and as a result, photosynthesis is not going to be able to occur, and this is what's going to cause the death of leaves in the fall. And lastly, ethylene, which is a gas hormone, believe it or not, and what this does is essentially causes ripening of fruits, maturation and aging. This is what's going to cause fruits to become really sweet. And a lot of times it does this by just breaking cell walls and allows that the juices containing the sugars to leak out. And it also contributes to the loss of leaves and death of flowers as well. So something interesting with ethylene is that a lot of the time you may purchase fruits from, maybe South America or Florida or like southern countries. And so obviously they don't want to purchase these fruits as they're ripening, because by the time they'd make it to Canada, they'd probably be, past their, ideal state. So what they can do is, they take these fruits and they're unripe still, but then they can kind of spray the fruit with this ethylene gas, and this is going to cause them to ripen so that when you put them in grocery stores, they can be ready for eating. And there you go. That is all about plant growth.